Click for IB Biology. Now we're going to look at what is actually controlling the cell cycle, all these different phases of prophase and metaphase, anaphase, interphase, and so forth. And it turns out it's the existence of some molecules called cyclins, which create checkpoints, trigger points within the cell cycle. There's a blank PDF of this presentation down in the description. There's a 10-question quiz with answers. There's also an AI prompt that will take you to a self-assessment multiple choice with any chatbot of your choice. Just copy and paste that in, and away you go. So let's take a look at this cyclin concept. This is Professor Tim Hunt. He was awarded the Nobel Prize for Physiology or Medicine in 2001. And Tim Hunt was studying sea urchins. He was interested in their reproductive behavior, and he had not set out to discover the control mechanism for the cell cycle. That was not his intention at all. But through close observation and analysis of data, much as you will do in your IA projects, Professor Hunt was able to make one of the great breakthroughs in biology, where you set out to work on one thing but discover something entirely different, is the phenomena called serendipity. So perhaps in your IA you also can experience serendipity. It's possible at all levels in biology, but Professor Hunt was a professional in his field. Let's find out what he discovered. So proteins, let's do P for protein, which have been phosphorylated, that is, they have a phosphate group attached, were known to be involved in triggering the stages of the cell cycle. This triggering are called checkpoints. But why the proteins became phosphorylated was unknown until the work of Professor Hunt. What he discovered was that there were a set of molecules called cyclins, and these would attach to a set of enzymes called kinases. So the cyclins would attach to the kinases, and in turn the cyclin-dependent kinase complex would result in the addition of phosphates to the regulatory proteins and activate those. And so then they would bring about the different stages of the cell cycle and indeed mitosis. It's worth mentioning that phosphatases actually dephosphorylate and return to the regulating proteins to their inactive state. So what were the patterns that Tim Hunt found? And so I've tried to summarize these here. We're going to focus in on only 4 of the cyclins, D, E, A, and B. And what he noticed in his data was that cyclin D began the increase in its concentration at the beginning of G1. So this initiated the G1 checkpoint, in which the cell began its preparation, its cell growth, its differentiation into its specialized function, and perhaps just the initial preparation for DNA replication. You can see that the cyclin D's concentration increases, reaches a peak, and then comes down at the end of telophase, and like so. So that's cyclin number 1. Cyclin D initiates the G1 checkpoint starts the first part of the interphase. Now the second cyclin that we're going to look at is called cyclin E, and notice that this has its peak of concentration in the S phase. So we think, what is it that's special about the S phase? And of course we know that this is DNA synthesis, DNA replication, and so we know that the increasing concentration of cyclin E in conjunction with the D increase brings about the G1S checkpoint of DNA replication. So that's this part of the interphase. So E is associated with S phase DNA replication. The second two cyclins that I wanted to mention were cyclin A. Cyclin A reaches its peak in G2, although it's always increasing from the beginning of G1. And cyclin A is associated with the G2 checkpoint. So if you remember what the G2 does, in G2, the cell has already done its S phase, it's already done its DNA replication, and now what it's going to do is it's going to do its organelle replication in preparation for the phases of mitosis itself. So this is a G2 checkpoint, the function of cyclin A. And finally, in our description, we're going to look at cyclin B. Notice that this reaches its pinnacle, its peak, in the metaphase of mitosis. And so we call this the cyclin B checkpoint, creates mitosis and the separation of the pairs of sister chromatids into their individual chromosomes, like so. Now, this has not exhausted all of the cyclins which have been found by Professor Hunt. There are more, but I've tried to choose the 4 most common and correlate those with the phases of the cell cycle as we have looked at before. So this is just a different image of the cell cycle with the peaks of the cyclins plotted onto the cyclic phase diagram for you. So the cell cycle is under the influence of regulatory proteins which have been phosphorylated, which in turn requires the activation of the kinases with the cyclins which are peaking at these points here as shown in the diagram. Now, this gives us the link to our next video, which is where the process of the regulation of the cell cycle goes wrong, and this results in tumor formation. There's the playlist link on the right hand side, the channel link in the center for you to subscribe. We're now going on to tumor formation in the last video in this sequence.